Matrix-assisted laser desorption, ionization, time-of-flight mass spectrometry, or MALDI, has revolutionized the clinical microbiology lab, enabling rapid, accurate, and cheap identification of bacteria, yeasts, and sometimes molds and mycobacteria. In most labs, it is used so frequently that it's become a verb. I MALDI'd it. But can it replace our antimicrobial susceptibility tests? Let's find out on today's Editors in Conversation. Welcome to Editors in Conversation. Please subscribe to this podcast and share with your friends and colleagues. This episode is brought to you by the Journal of Clinical Microbiology, available at asm.org/JCM. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. If you are a member of ASM, you can get up to 50% off the publication fees when you publish in JCM or any of the ASM journals. I'm JCM Editor-in-Chief Romney Humphreys, and I'm joined today by my co-host, Ellie Thiel. Ellie, good morning.
Good morning. You know, I love it when we can make new words in science. MALDI'd it, one of my favorites.
Yes, indeed. So today we're returning to my favorite topic, which is antimicrobial susceptibility testing. Ellie, I know you're thrilled about
I'm
this.
so thrilled. So thrilled.
So thrilled. Okay. And we are also joined by two guests. So first, Professor Frieder Schoenberg, who is a physician and director of the Institute of Medical Microbiology at the University of Munster in Germany. Welcome.
Thank you for having us today.
Wonderful. And we're also joined by Nicholas Weissman, a PhD student and biomedical scientist. Nicholas, welcome.
Yeah, thank you for having me here. I'm really happy to join the discussion.
Great. So we've invited you both here to discuss your recent publication in JCM, where you evaluated some machine learning models applied to our MALDI-TOF data to predict AMR. So this is something I feel like we've talked about since MALDI first came to market. It seemed like the promise of this expensive instrument to harness those spectra for strain typing, resistance mechanism prediction, virulence traits, all the things has been out there, but we haven't really seen it come to fruition. So maybe, Frieder, we can start with you. Why are we also interested in like turning our identification instruments into something beyond an ID instrument?
Well, MALDI-TOF, as you said, is a method that is applied by almost all labs around the world. And the beauty of this technology is that it is cheap and it can provide much more information in addition to strain typing and species identification. And therefore, we focus on this technology because we think and we believe that we can identify resistance mechanism within this mass spectra. And briefly, it is a cheap technology with a very short turnaround time. And the beauty is that we could maybe one day have species identification and antimicrobial susceptibility testing results at the same time.
Yeah, absolutely. That would be cool because it's so quick too, right? So Nicholas, maybe you can tell us a bit more about the types of approaches people have used to try to harness this data for AMR and the approach that you guys specifically used in your paper.
Sure. Let's speak broadly. There are two main approaches to harness MALDI data for AMR. The first idea is the direct detection of resistance proteins, looking for specific peaks, let's say PP2A in MRSA or beta-lactamase in gram-negative bacteria. But in our approach, we use data-driven machine learning rather than predefining peaks. We take the entire MALDI-TOF data spectrum, pre-process it and standardize in a standardized way, and let machine learning models learn patterns that correlate with resistance.
Mm-hmm.
In our study, we followed the Weiss et al. This is a paper which recently published, and we followed this approach very closely, and we did classic pre-processing with MALDI-Quant. This is an R package. Did some binding across the mass range of 2,000 to 20,000 and did some benchmarking in the end with several machine learning models. especially tree-based ones like random forest or gradient boosting machines.
So essentially, you're looking at the
Yes.
entire spectra that's captured for an identification with no preconceived notion of what in that spectrum may be driving resistance, and feeding that into a machine learning model to see if it can come up with something. So,
Yes.
and maybe just one more clarification: is this just predicting resistance, or does it predict susceptibility as well?
Yeah, right. This is a very important point. Technically, the model predicts both outcomes since they are trained as a binary classifier, so resistant versus susceptible. But conceptually and clinically, we should think of this primarily as a resistance prediction tool. The models are optimized to identify resistance isolates early. So far, this is not a replacement for standard AST. Rather, it could serve as an early triage for the decision supporting tools and would help guide empirical therapy while waiting for the lab confirmation, for example, the Vitek machine.
Got it.
So lots of potential here. But Frieder, what are the potential disadvantages of this approach?
Well, there are some disadvantage that comes with this approach. First of all, artificial intelligence and machine learning is a very new field in medical microbiology, and we have to be honest, at least if we speak for ourselves, we are not experts, and everybody is learning how to implement and use this technology. So expertise is really a key point, and I think we can only approach this new method and this new application if we have a interdisciplinary collaboration with data scientists, with machine learners, mathematicians, and so on. And I think that is very important to state. Nobody can do it on his or her own. And what we learned in our project is that we really need a lot of computing powers. A personal PC will never be able to manage all these analysis and models. That is more or less related to the infrastructure that we need to approach multi-mass spectra with machine learning tools. But the machine learning tools itself come with some disadvantages as well. As we showed in our work, we have to retrain our models regularly. And at the moment, although our results are promising, they can still be improved. And we are not yet here to use all these models and classifiers in routine care. So it's a research only use. And at the beginning.
Yeah.
And maybe finally, one has to state as well that all these predictions are based on a fixed drug-bug combination. So we have to use different models for different drug-bug combinations, and therefore it's not one size fits all, and that comes with some challenges as well.
Got it. So the computers are not yet taking over AST. Still need the human component.
Yeah,
still. So in your study, you set about to evaluate one of these potential disadvantages, specifically whether the data set used to train the algorithms, right, might negatively impact performance of AMR predictions when used by laboratories in other regions or later in time, with the idea being that AMR is regional and temporarily defined. So kind of diving into your study, can you describe the design? And Nicholas, this one is for you.
Yeah, thank you. Yeah, absolutely. Yeah, we built a large MALDI-TOF AMR data set over 2 years here in the University Hospital of Münster, and we focused on 3 major pathogens. This is E. coli, Klebsiella pneumonia, and Staphylococcus aureus. We trained multiple machine learning models and used repeated stratified train test splits with cross-validation to make sure our performance estimates were reliable. In simple terms, that means we split the data into training and testing sets, keeping the same proportion of resistant susceptible isolates in each. This helps us to see how the models perform on new and unseen data. So afterwards, firstly, We tested the reproducibility, so we applied the same machine learning algorithms to the giants data set, same as Weiss et al., and we received similar results. And in our second approach, we did some cross-site validation, where we trained our models on our Münster German data set and tested them on the Basel Swiss data set, and vice versa, to see how well the models generalize across geography and time. And in our third approach, we looked for the performance over time. We trained models on older isolates and tested them prospectively on samples collected over the following 18 months. That means we split this 18 months in 6 month intervals. This allowed us to evaluate how stable these models really are in real diagnostic settings.
Nice.
Cool. So let's talk a little bit about the stats, because I love a good stat. And so typically we talk about AMR in terms of, categorical agreement, very major errors, minor errors, which I realized recently, I heard that this is something that Clyde Thornsbury at the CDC invented. Anyhow, you all used a different approach, which was the area under the receiver operator curve for these assessments, but you also used something that was new to me, which was an area under the precision recall curve. And so I wonder, Frieder, if you want to explain that measure and why it was important to use it in your study.
Yeah, well, it's a similar approach that most of the people apply when doing similar analysis. And the reason why we applied the receiver operating curve was that the output of our models is the probability of resistance. So it's not that the model tells us it's susceptible or resistant. It tells us it is 60% resistant to ampicillin, for instance, in E. coli. And then the question that we cannot answer yet is 60% probability high enough to classify this isolate as resistant, or should we have a threshold at 90% probability? So we have a spectra of probability between 0 and 100%, but we cannot tell right now what is the right threshold to declare something as resistant. So therefore, we applied this receiver and operating curve where we changed the threshold and measured the sensitivity and specificity at different thresholds. And the result is such a receiver and operating curve. And this curve is good to show us how well can a model differentiate between a susceptible and resistant isolate. So it tells us, is it good to declare something as susceptible or resistant? And then we use this precision and recall curve because what we see in the receiver and operating curve is for some people only half of the truth. Because the performance of a test usually relies also on the background resistance rate. For instance, if you have 100% resistance, ampicillin resistance in E. coli, and you judge every isolate as resistant, you will have a perfect sensitivity to predict resistance without knowing anything. And therefore, this background information on resistance rates comes into play. And the precision and recall curve plots the positive predictive value for a resistant isolate versus the sensitivity. And as we probably know, is that this positive predictive value depends on the background prevalence of a certain feature, like resistance. We now consider the background resistance into this model. And a nice to know fact is that this precision and recall curve cannot drop below the true resistance rate that we have for the specific antimicrobial agent in our population. So both metrics show different aspects of the same performance, actually.
Actually, I kind of love that because, one thing in AST that I always find a little bit frustrating is, we classify things kind of binary. It's susceptible, it's resistant, but really it's more like, well, 80% sure it's resistant, or 80%, I mean, like, nothing is absolute. And so I do think that's a really cool approach.
Nice. Well, after that quick lesson in stats, I think we've sufficiently built up the anticipation of your study. So, Nicholas, tell us, what did you find? What was the performance of your model on your isolates?
Yeah, actually, it was quite good. In short.
Okay, we're done.
When we tested on the same site, we trained and tested on the same time, same site and the same time period. The OROC was above 0.8, which is classified as good performance. For example, in oxacillin resistance in S. aureus, or say fluoxacillin resistance in E. coli, or piperacillin tazobactam resistance in Klebsiella pneumonia. And what we also can say is that tree-based models consistently perform best, which is helpful because these models are much more interpretable than black box approaches like neural networks. For clinicians to trust the system in routine care, it's important they understand how predictions are made. And also what we can say is, for example, for the cross-site validation, our ROC dropped between 0.065 and 0.225. If not on the same site, which is also very impressive.
Pretty cool. Very cool. So follow up for you, Frieder. The best performing model was for ceftriaxone and cefotaxime for gram-negative rods, which had an area under the curve of 0.75 and the precision recall curve of around 0.29. So is that level appropriate for clinical testing? In my very, basic mind, it seems a little short of that 90% categorical agreement that the FDA and others, I guess, expect. So what are your thoughts?
Yeah, you are absolutely right. It looks promising, but it's not perfect at the moment. But we have to be sure and clear that these are two different metrics, area under the curve and these FDA acceptance limits. Coming back to your question, we cannot actually say which area under the curve corresponds to which FDA acceptable limit, because these are two different metrics. Maybe I can illustrate this on an example on Staph aureus that we did, but we did not publish it. Looking at Staphylococcus aureus and prediction of MRSA, we checked if we set the threshold how to interpret a predictive resistance from 0 to 100%. And if we increase this threshold from 0 to 100%, we see that obviously the very major error increase. So the very major error means the wrong susceptible ones increase while the major errors decrease. But there is no threshold in our data set that meets both requirements for the very major errors and major errors. But this is only one example, even if the area under the curve is quite good. And I think for Staph aureus and oxacillin resistance, it was around 80% or 0.8. It still does not meet the requirements to introduce a new diagnostic AST test. And yeah.
So do you think that should this proceed, do you think there would have to be some sort of kind of reset on the expectations of what is acceptable and what is not acceptable?
Oh, good question. I think the requirements still need to be high, because in the end we treat patients and we need to be sure that the treatment is correct. And what we see in other parts is that this category of major error and very major error becomes less important, and it's rather the correct identification of minimal inhibitory concentrations that play a more important role maybe in the future. And therefore, all these approaches that we are discussing today probably should not predict only susceptible or resistant isolates, but should also predict the MIC, which is even more challenging.
Yeah. Yeah, no kidding. Yeah. I feel like we can't even predict an MIC with an MIC test all that accurately. It'll be an easy target for you guys. I'm sure you'll figure it out. So, you guys, you trained your model on your isolates, and then you also looked at the model published by Weiss et al., and you kind of applied that to your isolates. So was there a change in performance? Because presumably they used a different bacterial population in their study.
Yeah, maybe I answer. Yeah,
absolutely.
First of all, this work by Weiss and colleague was really a groundbreaking work, and it was very inspiring for us. So we refer to Weiss and colleagues quite frequently in our work, and congratulations if they listen to this podcast. We used 3 models, Weiss and colleagues and ours. So logistic regression was one model, multi-layer perceptron, and light gradient boosting machine. And surprisingly, these models performed better on our collection than in the Swiss database. The reason why these models perform better in our database than the Swiss database is most likely that our database is larger or was larger, and therefore bigger opportunities for training, and resistance rates were higher in our database. So the likelihood to predict an isolate as resistant increases if the background resistance rate is higher. That is just a question of chance.
Yeah, interesting. And then Nicholas, the other thing you guys looked at was the predictive performance over time. And I was actually pretty surprised to see how quickly it changed. Like there was a shift even in a 6-month period. Did that surprise you, or did you kind of anticipate that you would see that?
Honestly, yes and no. On the one hand, we expected some performance decay over time because bacterial populations evolve and resistance patterns are shifting, but the speed of the decline was still striking, I would say, especially for Klebsiella pneumonia. And in some cases, we saw meaningful drops in our within just in 6 or 12 months, even though overall resistance rates were relatively stable. This is important to say also, because if there's a changing in resistance rates, you could assume that there's a drop in performance or a rising in performance, but if the resistance rate is stable, it's a bit surprising. This suggests that it's not just prevalence changing, but also underlying strain composition or proteomics patterns.
Yeah. I mean, like I said, I was really surprised by that because, like you mentioned, the percent susceptible doesn't change that much in that period, but I guess the strains do. I mean, you have different bugs moving in and out of your system. I find that fascinating.
So, Frieder, with that in mind, how should we be thinking about this? Should we all go buy some more MALDIs for our future AST testing on them?
Well, hopefully we don't need to buy more MALDI because we already use it.
True.
And we just extract additional information. But yes, I think MALDI-TOF based AST is our future. And because the beauty of having the identification and AST at the same time is just too tempting. But we are still far away from having it in routine diagnostics. I think for now we should see MALDI-TOF AST complementary to the regular MIC-based AST. For instance, we could consider MALDI-TOF AST as a quick and dirty method that we have at the same time when we do MALDI-TOF. And it should be combined for now with this slow but accurate MIC-based AST that comes usually 18 to 24 hours later.
Yeah, makes sense.
Yeah, and I think this was one of the things we had to ask you later, but I'll ask it now because we're on topic. Maybe the vision, as you say, is having the MALDI-TOF predict an early presumptive read, and then you finalize it with your final MIC testing. Yeah. So you can get a leg up if there's unexpected resistance, or maybe, a high probability of unexpected resistance.
So, Nicholas, just kind of going back a little bit. So if we have to update the model every 6 months or so, how many isolates do you need to feed into that? Or could the model be continually updated as the lab performs AST as part of kind of routine testing? So yeah, your thoughts.
Yes, you're absolutely right. Yeah, this would be the ideal scenario to implement the IST results immediately in the machine algorithm. But
Yeah.
this is technically, I would say, a lot of work, and you need the employees as well. So yeah, maybe in the future, but right now it's a bit too much.
Mm.
But that would be the ideal scenario, like to implement the IST results every day after the confirmation is in.
Got it.
Yeah.
And maybe I add some point. In fact, it is a continuous supervised learning system. If we feed the AST results into the system, and that would be really the perfect situation. This continuously learning supervised model.
Mm-hmm.
Based on your own isolates, right?
Exactly.
Yeah, absolutely. Cool. All right, Nicholas. So you're doing your PhD in this, so you're like deep into it. I'm curious, what other factors might impact performance that you didn't specifically study in this evaluation. So patient age groups, if the patient's in the ICU or not in the ICU, maybe even specimen types. Do you think that those types of things impact performance?
That's a great question, and we think they do. Actually, we are working on this and hopefully bring it in the next paper. We have another PhD student who is bringing all the environmental and patient data into our data set, and let's see how this will go on. We have a lot of ideas to bring in, for example, like prior antibiotic exposures or specimen types, also patient demographics, influence of lab values, for example, or other comorbidities, or even seasonal shifts. It's an idea. Travel activities. So a lot of different things you might consider.
Yeah, the sky's kind of the limit on it, isn't it?
Yeah.
All right. So I'll ask a final question. Are we essentially strain typing with this method? We know AMR is closely linked to the strain type. So CTXM and E. coli sequence type 131. Do you think the data go beyond that, or is this essentially like a strain type to predict resistance?
Well, I think you're absolutely right. In the end, it's more or less strain typing, because we see that some of our predictors are highly correlated. So certain peaks come in a group, and we don't know which peak is really predictive or which peak is really associated with the resistance mechanism, and which peak is really only correlated with this resistance mechanism. So I think what you say is most likely true, that we are typing and not really identifying the resistance mechanism. And to assess this basic research question, from my point of view, I think the best approach is to have isogenic mutants with and without this resistance mechanism, and then we can see if the prediction of resistance is the same or different.
Yeah. And I think back, when MALDI first came out and we were all very excited about this, I think, and I'll have to, we'll find it, folks, and we'll put it in the show notes, but there was a nice paper that looked at that for MRSA. And essentially they knocked out the MAC-A gene and the system still called it resistance. So, yeah. I mean, and of course it did, right? So, yeah.
Mm-hmm, mm-hmm. Well, this has been pretty cool. Nicholas, you're doing your PhD in diagnostic microbiology, which is awesome. Slightly envious, although I do not miss my PhD days. Share with us, what else are you working on? Anything exciting?
Yes, of course. As you all do, right?
Good answer.
Yeah.
Is that just because your boss is on?
Yeah.
Yeah, let's say first, I'm very lucky to be working on this in this field because, especially for me personally, it's relatively close to everyday clinical practice work. compared to basic research, and that's what's drives me even more. Beyond the study, what I'm working on is we are on our team focusing on improving the robustness and generalizability of MALDI-based AMR predictions. So we have more ideas to improve the pre-processing strategies, like new binding approaches. We have some ideas for linear binding, for exponential binding, or dynamic binding. Another idea is to do spectra calibration, to bring the spectra, like the peaks, more in line. There we also want to test differences in non-calibration and internal calibration. So internal means like all these peaks aligned to each other from the whole data set. But in external calibration, let's say that we align them to external reference peaks, which are well known. So the idea is in general that internal calibration may improve performance like within the same data set. But external calibration, we are hoping that this improves the performance like between different sites of data sets. That's the idea with the reference peaks.
That's awesome. Yeah, very fun. I wish I could geek out on all that stuff. That sounds great. Frieder, any last thoughts from you as we wrap things up?
Well, we are just grateful that we had the discussion today because it increases our visibility on this new approach. And as I said during this discussion, it is fundamental that we work together, not only microbiologists and microbiologists, but also across the different specialities. And we really need the support from machine learning specialists. And we have to open our doors worldwide.
Yeah. Absolutely.
I would agree with that, but I think our training programs also need to focus and dive in a little bit more and have a even basic understanding, right, of machine learning and algorithmic approaches and all that. So I think that's an area we can all improve on in our fellowships.
Exactly, and we should never trust the prediction models blindly, because we always need to understand what is really happening, and we need to check for, are the results plausible? Do they fit in the context? And therefore, machine learning tools are helpful, but we need to keep an hand on it, and we always should know what is really going on, and that's really important for us.
Just like using MALDI for identification. If it's calling something that doesn't make sense, then you got to question it. We still got to use our microbiology brains.
That's right.
Even if the score is good.
Yes, that's right. That's right.
Yes.
Well, thank you both for joining us. This was fascinating, a topic I love. Ellie knows this well. For those of you listening that are not as into AST, don't worry. I'm sure we'll be back to syphilis or something next month because Ellie's picking.
Favorite topic.
So relief is on the way if you don't love AST. But thank you all. And if you've enjoyed listening to this, please subscribe to the podcast and share it with your friends and colleagues. And thanks for listening to Editors in Conversation. As I mentioned, if you want to read the paper, which I encourage you to do, the link will be available in the show notes and at asm.org/EIC. Thanks so much and have a great day.
Thank you. Have a good weekend.